See. Here we go, warm up. All right, I'm gonna go share screen. All right. Yeah, one thing, if you have not been in a online class like this before, one of the weird things with the tests is, I hate it about the online tests, is it tells you what is your score when you're done, but sometimes people freak out because it gives them some score that looks horrible, but that doesn't actually mean that's your real score. That's like your score before I've gone through and manually, scored the things that are like essay questions. Also, it's before I've gone in and double checked. Like it has this thing where, if the right answer is, ribosome, and you wrote ribosomes, and I haven't like put every possible misspelling on, it'll say it's wrong. But if you wrote like ribosome, like ribosome or something, I'd probably give you credit for it. But it would say you've got it wrong for the initial grading. So, don't take the initial grades that it tells you too seriously, because that's before I've manually gone through and done the essay questions, and before I've gone through and made sure that there's not some problems with the short answers and stuff. All right, quiz statistics. Again, just to give you a sense, some people are getting perfect scores on these. Some people are really not. It's really important to try to keep up. Again, everything builds. All the stuff that we're talking about now, everything about these channels and gating and organelles, in fact, the channels are at the core of everything. All the signaling and the receptors we're going to talk about today are the core of everything. So you really need to make sure you keep up on this. All right, name at least 2 ways that an ion's channels in a cell membrane can be gated. So what are some of the ways you can control whether an ion channel is open or closed, that can open or, basically open the door or shut the door?
Voltage.
The one is voltage. We talked about how voltage is basically this charge separation across the membrane. Particularly excitable cells like neurons and muscle cells typically have this resting membrane voltage around -70 millivolts. We're going to be seeing some special voltage gated ion channels a little later this week that will stay closed if the voltage is -70, but if the voltage rises above -55, they'll open. And then if it drops down below, they'll close again. So we can have voltage gated channels. That's going to be critical in understanding how like action potentials are generated, which are the main way for long distance electrical communication in your body.
Professor, also, we're seeing some, or at least I'm seeing something different on your screen about the UC Berkeley neuroscientist versus the quiz.
That's okay. Well,
it's a very nice picture of Michael Silver, but
I'll show you. Okay, so I'll show you this. This is just announced. Yes. So Michael Silver is one of my best friends I've known since grad school, who I do the collaboration at Berkeley with the MDMA with. And he is now the director. They got over $1 million from an anonymous tech donor to start a psychedelic center at Berkeley. So this is, I can, in fact, I'll put this in the chat. You can, yeah, it's really, really exciting. It's kind of amazing that this actually happened. So he's there, and he's like, I don't know if you know Michael Pollan, he's this author. He's thrilled to be working with him. He says he's a really, really cool dude. But anyway, you should check this out. This is another, David Presti's another old friend of mine who teaches the drugs in the brain class at Berkeley. But it's exciting. It's very exciting to actually have this all happening right now. So I'll put this link in the chat if you want to read more about it. So but that is not what I meant to be sharing there. Thank you for checking that out. All right, let's see. Boom, this screen. That looks like it. All right. And I do want to, I'll put this, everyone. All right. So what other ways besides voltage do we have for gating a channel?
Mechanically gated?
Mechanically gated. So just by stretching the cell, stretching the membrane. Again, that's going to be how we end up having touch receptors. That's actually how you hear me, is the air vibrations are bending little cells and creating channels opening or closing, ions moving across the membrane and making voltage changes. What other ways? Ligand gated. So chemical messengers come in. For instance, like neurotransmitters or hormones can come and bind and cause channels to open. So those are all going to be important. Next, in secondary active transport. So most people, we have 88% of the people, solute moves against its concentration gradient using the energy stored in, so that's good. Most people got it. Particularly if something's called active transport, it's probably not passive. Passive transport is diffusion or facilitated diffusion, where things are moving along their concentration gradient. And definitely not stored, not using energy directly from ATP. That would be primary active transport. You use primary active transport to set up the concentration gradient in this other solute that then drives the secondary active transport. Next. This is where I try to put in different answers that people will put in. So ribosome, cell organelles at their site of protein assembly are the ribosome. What are the organelles that are the site of ATP production?
Mitochondria.
And we're going to be spending a lot of time looking at the nitty-gritty details inside the mitochondria this week. If a cell had a membrane voltage of minus 70 millivolts, so remember the voltage is the inside compared to the outside. If we are considering just the force due to electrical potential, a cation such as sodium will want to move in or out of the cell if it's minus 70 millivolts inside the cell compared to the outside.
Into.
Going to want to move in. Remember, because opposites attract. If you're a cation, you want to move towards the more negative. If you were an anion, a chloride, for instance, that would want to move out to where it's more positive outside the cell. So this is going to be also important. Probably this week, we're going to be talking about how the resting membrane potentials are set up, and it's going to be a mix. Partly ions, up to now, we've been talking about how things move due to diffusion gradients, right? If I have a whole lot of, I could think about sodium moving due to diffusion. That's what we've been talking about right now. There's a lot of sodium here and not much out here. It's going to move from high to low by diffusion, the chemical force, basically. But then there's this other thing that's going to come into play, particularly when we're talking about ions, is this electrical force, which might be in the same direction or might be in the opposite direction as the diffusion force. So we're going to actually find these equilibriums where they fight each other and find a balance. That's how cells are going to reach their resting potential, actually, is going to be this balance where Diffusion wants it to move one way. The electrical force is building up, pushing back the other way, and it finds some equilibrium. And finally, choose all that are true about facilitated diffusion. So this people got kind of clobbered on this one. Most people knows that it requires carriers. Most people know. It is a form of active transport. It is not, even though it sounds, you know, that one, it sounds like it's facilitated, but it's passive. It's passive transport diffusion through a carrier. The difference between regular diffusion and facilitated diffusion is there's a rate limit, which 20 people got. But so wrong, no rate limit. Does not require ATP because it's passive transport. And it is a form of passive transport. So that's a quiz. Is there any question about any of the warm-up questions?
I have one question, Dr. Egger. For the last one, I thought it used ATP from the pump, or is that wrong?
No, facilitated diffusion is purely just diffusion using a carrier to cross the membrane.
I think I'm thinking of the other question. Was it the secondary something? Is that the one which one uses the pump?
Secondary active transport, there's a pump that is used for, you know, in the background to create the concentration gradient of the other solute, but the actual driving of our solute is done by a symport, where like I showed how like the sodium came in and pulled the glucose in the transepithelial transport.
Okay, thank you.
Sure. Okay, let's see.
I did have a question in regards to the voltage. So I had noted down that these cells typically at rest at -70, but you're saying that's when ions would be passing through, is when it's at rest?
No, it's a cell at rest is typically around -70 millivolts, and that's when things are kind of in an equilibrium. So that's where this kind of diffusion force and chemical force and electrical force are in balance. Kind of like in osmosis. In isotonic solution, there's no net movement of water in or out of the cell. In a cell at rest, there's no net change in the charge separation across the membrane.
So I guess I got confused with that on that question with the cell membrane being at -70.
So what is your question?
Because I thought that was the cell at rest.
But here I'm saying it will tend to. It says just considering the force due to the electrical potential. In a real cell, it's not just electrical. There's also that diffusion force because there's a major difference in concentration of the ions inside and outside of the cell. So a cell at rest, at resting potential, there will be no net movement. But here I'm saying, this is this artificial thing. It says we are just considering the force due to the electrical potential. And if we're just thinking about electrical potential, it's strongly pulling the sodium into the cell. So, does that clarify things?
Yeah, thank you.
Okay. Okie dokie. All right, so let's continue to dig in. We've got lots more to cover about the cell membrane and the cell membrane proteins. So let's do it. I'm gonna back to the whiteboard. Okay. So membrane proteins. continued. Again, we have our cell. We've got all sorts of membrane proteins. Some of them go all the way through. Some might just be on one side. And we talked about some of the functions of these membrane proteins. What are functions of these membrane proteins?
Structure.
So, in fact, we're gonna. Let's continue with the one that we've been working on. What's one of the one that we're kind of in the middle of?
Transport.
So we've been talking about transport. So transport, we talked about there are channels, basically things that can let ions in or out, and these can be gated. We talked about carriers. Yeah, it's kind of more complex, usually for larger, more complicated things. These are usually for, like glucose. Sugars have to use carriers to get in and out of the cell. Things like amino acids need to use carriers to get in and out of the cell. Things still kind of follow their diffusion gradient, but they're rate limited. That's where we get facilitated diffusion. We talked about those co-transporters. Basically the symports and the antiports. Sometimes things have to move in together. Sometimes things move opposite. A little later in lab today, we're gonna meet an antiport you find in the red blood cells that's gonna explain some of the weirdness that we're gonna see in our results. Some of these can be pumps using ATP, where you actually have the primary active transport. So we've got to meet all these different forms of membrane proteins that are going to be critical for our semester, talking about how things get in and out of a cell, how you control what's moving in and out of a cell. Since we're at transport, I should mention one more form of getting things in and out of the cell before we move on to the next thing. What's another way to get things in and out of a cell? Obviously things can just diffuse if they are penetrating, like urea or something. They can move in through a carrier or a channel if there is a special membrane protein. What's the one other way we can get things in or out of the cell that's not using transport proteins and not just penetrating through the membrane?
Endocytosis.
Say it again.
Endocytosis.
Yeah, exactly. Did everybody hear Jamba? Endo and exocytosis. So now would be a form of active transport. Because this takes a lot of energy and kind of machinery to make this run. Here, the classic thing, we have our cell, and let's say here's some little thing that you want to take in. Through the miracle of time-lapse photography, we can just show how the membrane kind of moves around it, brings it in. This would be endocytosis if you're bringing it in. We could also do it the other way. If you have something that's stored in little vesicles, you can spit it out. That's exocytosis. So this is how you can bring things into a cell. It can also be, this is how we're going to release neurotransmitters and hormones and stuff. You make the neurotransmitters, they're stored in little vesicles, and you do exocytosis, and now they are being spit out in the synapse of the between the neurons. Another thing you can do with endo and exocytosis is actually not even use it to take things in or out, but just to change what kind of membrane proteins are there. For instance, let's say here's my cell. Like when we get to the kidneys much later, we're going to talk about how the collecting ducts can be very permeable to water or not very permeable to water, depending on the presence or absence of ADH, the antidiuretic hormone. So let's say I have a whole bunch of like aquaporins, little water channels that make this permeable to water, but now I don't want it permeable to water. What I can do is I can do endocytosis. And in a fraction of a second, now there are no water channels in the membrane to make this permeable to water. And in a fraction of a second, I can go the other way and I can refuse this little vesicle part of this little membrane part, and all of a sudden they're back. So this is a way to very quickly and easily change the membrane properties of a cell, because you can add and subtract membrane proteins that are currently being stored in the membrane of this little vesicle. I should also just mention, when I'm drawing it like this, it just looks like magic. I'm just drawing it. It's like, oh, it's sucking in. And obviously this is happening with all sorts of machinery. This is mechanical. People often talk about clathrin-coated pits. If I have a cell, it's not just any random part of the cell that is being the place that's doing endo and exocytosis. You have a little place like we call like a clathrin-coated pit. Clathrin is another membrane protein, and it's actually super cool. It's got this like kind of trifold symmetry, and what happens is when that little segment of the membrane comes in, the clathrin actually locks in around and creates a scaffolding that stabilizes the vesicle so it stays intact as a little bubble inside the cell. So the area, so that's just an example. And obviously there's got to be all sorts of cytoskeletal elements pushing and pulling to make this happen, right? Membrane is just like phospholipid. It's not going to push in or pull out, without some help, something pushing and pulling on it. So all of this, that's why I'm saying this is a type of active transport to get things in and out of the cell, because it's going to require a lot of machinery and energy to manipulate the membrane and make this all happen. Obviously, once the membrane pinches off, because phospholipids are just held together by hydrophilic interactions, it'll just automatically seal off. You don't have to do anything special when you pinch off a phospholipid bubble, because the membrane, spontaneously forms into the bilayer again. But yeah, so endo and exocytosis are going to be important. But back here. So membrane proteins, transport. I think at this point we have covered the main transport. Now we can talk about what's another type of membrane protein that we got. So 1, transport. 2, what's the other one?
Structural?
The structural. So I'm going to say, kind of cytoskeletal attachment. I realized actually when we did our description of organelles, I didn't actually talk about cytoskeleton, so we should do a little detour. Again, a cell is not just a little Ziploc full of goo. It's got a very highly kind of organized system of scaffolding, and things are anchored in place, and things can move along little railways to get pulled from one side to the other. What are different elements of a cytoskeleton? There's like 3 main cytoskeletal elements we usually talk about.
Are tubules, microtubules?
The one is microtubules. So microtubules, these are made out of these little tubulin subunits. So microtubules have these little subunits, and you can add them in and lengthen them. You can get rid of them and shorten them. Microtubules are what makes up the spindle in mitosis, right? The things that reach out, grab on to the kinetochores of the chromosomes and pull the sister chromatids apart. You know, those are microtubules shortening. They're getting long as they grow in. They're shortening as they go through anaphase and pull things out. Microtubules are also, when we get to neurons, with these big long cells that might be 3 feet long, microtubules are going to actually be like little railways where you can drag things along from one side of the cell to the other. Microtubules are also really important for cilia. I'll get to the cilia in a second. Let's finish the different cytoskeletal things, and then I should talk about cilia briefly. Okay, microtubules. What's another kind?
Centrioles?
So the centrioles are like the microtubule organizing centers.
Ah, okay, okay.
Centrioles are the things that
Actin.
are kind of building the, so actin, which we often call microfilaments. Actin, actin is what holds, you think of the nucleus as in the center of a cell. Why is it staying there in the center? There's like actin holding it in place. Actin is going to also be at the core of a lot of the kind of contractile properties when things squeeze and pull. Actin is going to be what's strengthening the little microvilli I'll talk about in a few moments. And then there's a catch-all that we use, intermediate filaments. Intermediate filaments just as kind of everything else, like in skin, it was a keratin, things like that. But obviously the cytoskeleton is this thing helping it hold its shape, and there has to be attachment points onto the membrane. Otherwise we couldn't have endo and exocytosis if you didn't have structural elements from the inside pushing and pulling on the membrane. It's how you have a columnar cell stay columnar looking, and a squamous cell stay squamous looking, right? It's how you have a red blood cell stay red blood cell looking. Is you've got a cytoskeleton, and then the membrane is, anchored onto the cytoskeleton. So cytoskeletal attachment is another important role. I am thinking, because it's worth talking about cilia and microvilli, let's just do that right now. So a slight tangent. Back. So different, we could have our kind of classic cell that just looks like this. And the membrane is just kind of smooth. But then we have a lot of different options. One is we can have the membrane really folded up. We call microvilli. And these are reinforced with actin. Right, the actin, think about, like, I've got this nice shirt here, and I got the collars, and the collars stay kind of nice and kind of crisp because there is interfacing. There's this kind of stiffer material that's sewn into there so it holds its shape. So the actin is kind of like the interfacing in a collar that keeps your collar nice and sharp. It's basic, because otherwise, think about if you just took some cloth and you pleated it up with little folds and let go, all the folds would just fall out. So we need to have cytoskeletal components that help it hold its shape. The microvilli, these are submicroscopic. These are only visible distinctly under an electron microscope. And when you look at these, if you've been in anatomy, you kind of know, we talk about kind of the brush border. Again, when we look at the intestines, for instance, you don't actually see you can tell that something's going on. This is more what it looks like in a real microscope, but this is basically going to very much increase your surface area. Right, so if you think about, if I have a certain amount of surface area, to absorb, remember we talked about how surface area affects rate of diffusion. So if I take the same amount of surface area, but now I kind of squish it down, I have just as much surface area, but it's more compact. It takes less room. So by using microvilli amongst other tricks, you're going to have a surface area about the size of a football field to absorb nutrients in your small intestine. So microvilli, they are not motile. I mean, they don't move. Not motile, but they are super important for increasing surface area. And again, you usually find them where you want to efficiently absorb things across the membrane, because by increasing the surface area, you're going to increase the rate that things can cross, as we saw in our lab last week. The other thing, it looks kind of similar, but is not, are cilia. So these do move. Cilia are, all right, let me put these on another page. Motile. These are going to be able to sweep things across the top of the cell. These are the things that sweep the egg down the fallopian tube after it's been ovulated as it goes down towards the uterus. These are the things that are sweeping the mucus up your respiratory tract so you can get rid of it after it's trapped the dust that you've breathed in, particularly these days. So So they sweep things across. These have microtubules as their, so these have microtubules. They have these things called basal bodies, are the organizing centers for them. And we have what's called the 9+2 arrangement of microtubules. What that means is if I take like a cross section through one of these things, so this is just supposed to be one finger here. I'm going to have, in the center is a pair, that's the 2, and then the 9 is going to be 1, 2, 3, 4, 5, 6, 7, 8, 9 pairs of microtubules that are around that. And then there are these special little proteins that link in between, little dynin proteins. Dynin sound, dynamic motion. So imagine I've got kind of the center and I've got the microtubules on the side. And then I have these little dynin proteins that pull these two. So in fact, where is it? I've been leaving this thing on my desk for like the last few days, so I would have it here. And wait, when I'm looking for it, it's gone. Oh, here it is. Okay. So I'm gonna stop share for a second so you can really see. So what I'm holding in front of me here, this is actually the top of a Ziploc. So there's 2 parts to it. This is so easier if I stop the virtual background for a moment. So here you can see this thing, there's 2 sides. I'm going to Ziploc them together so they are held tightly. They have to move held side by side, but they can still slide. So then I am going to use my fingers to create a force on the bottom that's going to make them slide past each other. And you can see what happens. It just starts waving back and forth. That's kind of what your microtubules are doing in the cilia. They are getting pulled up and down side by side, and it's creating this flexing of the whole little cilium. So that's how they work. It's just those microtubules, the little dynin proteins pulling, and as they get pulled, it makes the whole thing flex one way or flex the other way. Does that, hopefully that makes sense. Again, this is like one of the classic things you can do if you have like if you have a Ziploc and you have kids around, you can take the top of the Ziploc off. This is just the top of a Ziploc. Usually you want to you have to break the seal on the bottom part, and then you can just hold it and use your thumbs and your forefinger, and then you can do kind of magic tricks. You can say, I'm going to rub it and make it all charged. And now I'm going to pull my hand away and look, and then I'm just going to stare at it and make it scared and it runs away. So that's something you can do when you can make up whatever kind of story you have. You can have oh, look, it wants to be friends with the pen or whatever. You can, so have fun with it. Anyway, let's get back. So cilia, motile. Again, we'll see them. I mean, there's one related thing, which is a flagellum. It's the same basic idea, but instead of waving little hairs that sweep things across the cell, this whole thing will wave back and forth and propel the whole cell through space. In an adult, these only exist in one place. Where's the only place we see flagella in an adult?
Sperm.
Sperm. During development, there actually are flagella that are spinning. You know, if they don't spin right, it actually messes with the differentiation of things. They spin the opposite way. I think even you can get all your organs on the wrong sides and stuff. But as an adult, only the sperm have flagella. So there's one other thing called stereocilia that we'll talk about that are basically like long microvilli, but let's not worry about them now. Okay, so we've been talking about membrane proteins. We said transport. Check. Cytoskeletal attachment. Check. Where's some more? More functions of membrane proteins.
Is this cell ID?
Yeah, definitely. Cell identification. So we're going to talk about this in much more detail when we get to the immune system, but your body needs to have a way to know if a cell belongs to be part of you or if it's some invader. So every cell in your body, with a few exceptions, have these special things, the major histocompatibility complex proteins that have a very specific genetic marker that only would show up on your cells. You know, that's why trying to, even if you desperately needed a kidney to stay alive and I desperately wanted to give you my kidney, the chances are it wouldn't work because your body would say, these cells look wrong. They don't have the right ID markers on them and I'm going to attack them. And that's when you have like the graft versus host disease, a transplant rejection, because each one of your cells has a very specific cell ID marker on them. And unless you're genetically very close to the donor, your body's going to have a very strong immune response to attack and get rid of that. Even if it's a very close genetic match, you still are going to be on anti or immunosuppressant drugs for the rest of your life so you don't, you know, take out your donated organ. So cell ID. What other things are membrane proteins going to do?
Cell-to-cell attachment.
Cell-to-cell attachment. Right, you've got all of these cells, trillions of cells, but they're all stuck together to make a human being. Some of them are just kind of held together by these kind of gluey things, proteoglycans and stuff that kind of just make things kind of stick together. But in other cases, there are very specific proteins in the membranes that hold one cell attached to the cell next to it. There's 3 particular versions of these you need to know about. So let's do our drill down into cell-to-cell attachment. One version are called tight junctions. These, they're, again, I think of them, you were talking about Ziplocs. These are kind of like Ziplocs. So when we talked about the transepithelial transport, again, hopefully all of you totally understand this. If you do not understand all the details of this, you need to make sure you get up to speed on it. Remember, I had my epithelial sheet. I had sodium and glucose coming in, and sodium getting pumped out here, and glucose coming out here, and all this stuff. The whole point here was I wanted to be very intentional about making sure that I'm pulling my glucose into here, but I don't want other things. I want to only pull things across that I want to absorb. Other stuff's going to continue out and become my poop, right? I don't want things accidentally going back out that I've just taken in. So I need to make sure that the only things moving across in general are the things that are being intentionally moved with all these different processes, which means I need to seal the gaps in between the adjacent cells. And that's what the tight junctions are. The tight junctions are kind of these, kind of like a Ziploc between the adjacent cells, so things cannot wiggle through. If something is trying to get through, it can't make it because the Ziploc, that the membranes are basically folded in on each other with these special proteins. So the only things that are going to move from one side of the epithelial sheet to the other are things that are being intentionally moved. So does that make sense? Tight junctions, like this whole system here of transepithelial transport wouldn't actually work if you didn't have the tight junctions, because otherwise things would just move, you know, just wiggling, getting around the back end, right?
So are tight junctions types of proteins?
They are proteins. They're proteins that are in the membrane and then kind of lock one cell's membrane onto the other cell's membrane. Exactly. Another kind of cell-to-cell attachment that we will be seeing in this class are the gap junctions. So I have my cells, and gap junctions literally connect the cytoplasm from one cell to the other. These are little, they call them connexons. They play many different roles. The main thing that we're going to be seeing them do in our class is they allow ions to move freely from one cell to another cell. What is that going to mean if ions can flow?
Electrically charged.
It means there's electrical current. So this electrically connects the cells. You know, there are other functions for gap junctions, but in particular, as we go through the semester, this is the thing we'll be particularly paying attention to. If we see cells that are connected with gap junctions, like when we get to cardiac muscle or smooth muscle or electrical synapses in the nervous system, if you, or in the heart, we'll see this as well. If you excite one of the cells, the next cell will automatically get excited because the electrical current will automatically flow to the next cell as well. So gap junctions, make sure they electrically connect cells. You know, and that's because ions can flow back and forth, as well as other things, obviously. Fine, then 3, just for completeness, let's use desmosomes. These are more structural. These are like, I talk about them being kind of like rivets and cables. You know, for instance, in cardiac muscle or in your skin, there's things where you have things under a lot of tension and you don't want the cells to rip apart. You don't want your cardiac muscle cells to pull apart from each other as your ventricles are squeezing really hard. You don't want your skin cells to all come apart from each other. You're scratching and all of a sudden you've dug a hole in the side of your face. That'd be gross. So what you have are these membrane proteins that are these mechanical, they're very structural. They kind of mechanically anchor things together. They usually have fibers that then reach in and anchor into the cell. There's often filaments that actually move across the entire cell from one to the next. So it really anchors these cells. So even if they're under a lot of force, they're not going to rip apart from each other. So this is basically, you know, structural integrity. So when we have desmosomes, that's keeping cells from getting pulled apart, even if they're getting, you know, pulled hard. So my gosh, cell-to-cell attachment. But wait, there's more.
Phagocytosis.
So I would say cytoskeletal attachment would be, that would be under here, because you're basically using cytoskeletal elements to push and pull. And then there is also the clathrin that I talked about, but I've taught, you know, we won't put it on here. I could put it, clathrin is also up here. Let me be less quick. So I will put that on the side, but it's not going to be a major player for our semester in terms of what you need to understand. What are some other ones?
Is communication one?
Yeah, a big part is, so again, you've got like 10 trillion cells and they all have to be able to know what the other ones are doing. You know, you have to have cells be able to like talk to each other. You have to have cells know what's going on outside. You have to have cells know, like, are there nutrients around? Are there toxic things I need to get rid of? Are there things I want to take in? Or so this whole idea of receptors. And this is something we're going to talk about in a lot of detail in this class. We're going to get into some of the nitty gritty of receptors down to like the, you know, protein structure and how the different side chains of the amino acids actually interact with the, like a neurotransmitter like serotonin or something. So we're going to talk a lot about receptors, and we're also going to talk about all the second messenger signaling that gets activated when a receptor does its binding. But let's start with an introduction to receptors. So here's my cell. Here's some receptor protein. So the receptor protein. And then there's always going to be some kind of a ligand that binds to the receptor. Basically a signaling molecule. So the ligand, it could be a neurotransmitter, it could be serotonin or dopamine or something binding a receptor on a neuron. The ligand could be insulin binding the insulin receptors on your cells, saying that there's sugar in your bloodstream. The ligand could just be sugar in your saliva that's binding onto the receptor on your taste bud, right? Ligand is just a molecule. It's just a molecule that is going to bind to the receptor. The binding is similar to what we talked about when we talked about receptor and substrate binding, in that it's very kind of shape specific. Shape and chemical property specific. So if you have a receptor that binds serotonin, and then you have a dopamine, the dopamine isn't going to have the right shape or chemical structure to bind. So it's not going to, only things that have a very specific shape and chemical structure are going to bind and activate this thing. That being said, it is possible to fool it by giving it things that like mimic the naturally occurring ligands. So let's talk about a couple of things. First, let's talk about binding affinity. And then we're going to talk about exogenous and endogenous ligands. So binding affinity. Binding affinity is how tightly the ligand holds on to the receptor before it lets go again. Right? Obviously, when a ligand binds the receptor, it doesn't get stuck there. That would be like breaking the keys off in the lock, and then you'd never use the door again, right? So the ligand binds the receptor, but things are, again, temperature, things jiggle and it lets go. And if it's still around, maybe it binds again, and then it breaks off, and then it lets go, and then it comes in. Eventually, probably it gets cleared out by some physiological processes. Some ligands tend to, once they get on, hold on much tighter before they let go. Some of them grab on, but they can get knocked off very easily. So binding affinity is saying how, if it's high affinity, that means it holds on tight, it's harder to knock it off. Low affinity means it will grab, but then it gets knocked off pretty easily. We're going to see that it's going to play very much into like dosages of drugs and things. So we'll talk about that in just a few moments. So now we're going to introduce the idea of endogenous versus exogenous ligands. So endogenous, endo, within, created within. These are the naturally occurring ligands that are in your body, like insulin, serotonin, just whatever the normally occurring thing that's part of your physiological processes. Exogenous, created without. So these are from outside, but they have a shape and chemical properties that are substantially similar enough to an endogenous ligand that they can bind the receptor. For instance, like albuterol. If you have asthma, you take your little albuterol inhaler. that is an exogenous ligand that has a shape close enough that it can bind adrenaline receptors and help dilate your bronchial passages and make it easy to breathe. So does this idea make sense? An exogenous ligand? It's something that is basically like a mimic. It's a mimic. It's close enough that it fools the receptor into binding.
One question, Professor. For example, some medicines that we take are ligands, antidepressants or insulin when a diabetic person has to.
Right. So insulin would actually be the actual thing that the receptor.
Okay.
For instance, we'll talk about, like, albuterol is something that isn't quite adrenaline, but it will still bind that receptor.
Okay. Okay.
And I'm going to, so let's talk about antagonists versus agonists. Actually, let's start with agonists. So just because you bind the receptor doesn't necessarily mean you're going to activate it normally. So if I have an exogenous ligand and it binds the receptor and it activates it, we say that's an agonist. So I can use a type tool, probably be quicker. Example of this would be like morphine or heroin or any of or Vicodin, hydrocodone or oxycodone, any of those opiates. They can get into those opioid receptors, they bind it and they turn it on, which helps with pain relief and makes you feel floaty and all that. So that would be an agonist, an opioid agonist, something that binds the opioid receptor and activates it. An antagonist, on the other hand, I should say binds receptor, but does not activate, but does block it. So an exogenous ligand, something that will bind the receptor, it doesn't turn it on, but it doesn't let other things attach there to turn it on. So an example of this would be like beta blockers, like beta adrenergic antagonists. Like people take beta blockers to try to keep their heart rate down if they have high blood pressure, or actually if they're stressed because they have to give a talk or something. You take a beta blocker, something that binds and blocks adrenaline receptors. It means that all of a sudden you're scared and you get an adrenaline dump in your body, but the adrenaline can't get at the receptors, so your heart just doesn't speed up and you don't feel as freaked out by things. So an antagonist binds the receptor, but it does not activate it and it blocks it. A classic, in the medical field, a really important antagonist, in fact, something that will give you a sense of affinity and all this, Narcan. Most people, does everybody know what Narcan is?
It reverses the effects of opioids for like 20 minutes, doesn't it?
So this is Narcan, naloxone. If you have somebody that has an opiate overdose, like if you take too much of an opiate, it can be really dangerous because it'll suppress your breathing and you can die. So you really you need to get in there and you need to turn off the effect of the opioid before the person dies. So Narcan, this is a very high affinity opioid antagonist. So let's say here's the cell of our person who's OD'd. And here are the little opioid receptors. And let's say here's all the little heroin molecules that are kind of in there that are activating. And what we need to do is we need to try to get these out of there and stop activating these receptors. This is our little opioid receptor here. You give somebody Narcan. Narcan comes in. Narcan is an antagonist. It's going to actually get in here and it will bind. And it's high affinity, which means it really wants to hold on. So the little heroin molecules are still hanging out here. There's still a bunch of heroin, but the Narcan isn't letting go. It's not giving the heroin a chance to get into the receptor and bind. Because once the heroin gets in and, in and out and in and out, but when it goes out, the Narcan comes in and it just stays. So Narcan will let this person be all right until the body naturally clears this out. So you have to make sure the Narcan stays high enough in their body until the body has had a chance to clear the other opioid out of their body. But yeah, the people I've talked to people who've administered Narcan, and it's supposed to be pretty dramatic. A lot of times the people get really angry and pissed at the person who gave it to them because they've just been in this, beautiful, transcendent, floaty place, and all of a sudden they're just back here in this horrible place and get really pissed, like, what have you done to me here? But they've saved their lives. But yeah, people can get kind of belligerent when they first come back because it's just such a quick yank back out of the space. Again, this is becoming more and more commonly a part of a normal med kit, just to try to save somebody who has an opiate overdose. Does this make sense? This idea of, an agonist, again, morphine or Vicodin or oxycodone or heroin, those are all opioid agonists. They're things that will bind the opioid receptor and activate it, turn it on. Antagonists, things that will bind, but they don't activate, they block. Again, they're really useful as well medically. Again, we have like adrenergic agonists, things that block adrenaline receptors, like to keep your heart rate down, for instance. Or you might want an adrenaline agonist, again, for asthma, because part of the sympathetic activation is to dilate your bronchial passages. Affinity can also affect dosage. So if you think about like morphine versus fentanyl, both morphine and fentanyl are used, clinically all the time, as, for pain. The dosages are really, really different. Fentanyl is very high affinity. Meaning once fentanyl gets to the opioid receptor, it holds on and keeps activating it for quite a while before it breaks free. Morphine grabs on, breaks free, grabs on, breaks free. A normal dosage of morphine, you get wheeled into the ER or something, they'll give you like maybe 30 mgs of morphine. For fentanyl, you literally need 100 micrograms. That's Mike, right, this is 0.1 milligram. So this is 1/300th as lower dose than we have for the morphine. Right, so it's a huge difference in how much you actually need to be effective in the body, because the fentanyl just is so much better at holding on to the opioid receptor and keeping it activated. Similar thing, if you think about like classic psychedelics, like the classic psychedelic like peyote and stuff, you know, mescaline. Mescaline, you might need like maybe 250 milligrams of mescaline to have a proper trip, like psychedelic journey with mescaline, with a peyote or something. With LSD, again, it's another thing with incredibly high affinity. You just need, again, about 100 micrograms and you are solidly in that psychedelic space. So again, this is a difference of 2,500 times more mescaline that you need by weight than LSD to have the same effect. You know, and it has a lot to do with affinity. So this idea of affinity can definitely affect dosage. We saw in terms of agonists and antagonists, it can affect, you know, just which one is winning in the battle for the receptor, which can be really important. A lot of drugs that we are going to see are either agonists or antagonists, or sometimes partial agonists, whether they are clinical pharmaceuticals or recreational drugs. Again, if you think about THC, why do people get stoned? You know, it has a shape that binds your anandamide receptors on your neurons. You know, why does caffeine keep you awake? You know, this is a adenosine antagonist. It turns out adenosine, in addition to everything else, is used as a neurotransmitter in your brain, and it builds up as your neurons keep working, and as it gets higher and higher levels, it binds adenosine receptors that are part of the circuitry in your brain saying you should sleep and rejuvenate. So if you give somebody an antagonist to those receptors, then your brain isn't getting the message that adenosine is building up and I need to sleep, so you feel more awake, right? Did I misspell caffeine? I don't remember how to spell caffeine now. Whatever. I think I spelled it right. Which other ones? Benzodiazepines. These are like Xanax, Valium, etc. You know, these are something a little different. These things will actually bind, but they don't bind at normal receptor sites. They actually change the behavior of different channels. So these are a little different, but they're still related enough that I'll introduce them now, and I'll talk more about them later. When we get into the GABA receptors and the chloride channels and stuff, we'll talk about how you can actually attach to these things and actually change behaviors to the native ligands. So benzos will actually make the normal functioning, increase the normal functioning of the native endogenous ligand in ways that we'll talk about. Other things, you know, all the psychedelics, LSD, psilocybin, mescaline, these are all agonists that look like serotonin enough that they bind serotonin receptors and do this partial activation of them. There's all sorts of agonists and antagonists for the sympathetic and parasympathetic neurotransmitters. You know, nicotine. Why does nicotine wake you up and focus you? It is an agonist to acetylcholine receptors, which are part of your attentional circuits. So nicotine is just another agonist to a receptor in your brain. If you want to block, let's say you get an eye exam and they shine a light into your eye. You know, if you shine a light into someone's eye, normally your pupil constricts. But if you're in an eye exam, you want the eye to stay open so the doc can look in there and see what's going on. So you give them an antagonist to acetylcholine, which activates the muscles that constrict your pupils. So you might use an antagonist to just keep your pupillary constriction reflex from operating during an eye exam. So these drugs are used all sorts of ways, clinically, recreationally. But again, the take-home message is this idea of agonists can bind and activate, antagonists bind and block. And then there are things that are in the middle. There are things that are called partial agonists and stuff, but we'll talk about that later. Any comments, questions, or anything about all this? Wendy? I can't hear you.
Hi, can you hear me?
Okay, yeah.
Okay, sorry. I have a question about the fentanyl. I've heard a lot of like first responders when they get a call to help someone out, they usually get like transdermal. It'll like go and affect them.
Yeah.
Is that still due to the like high affinity?
It's because right. So fentanyl gets a lot of in fact, that was just that thing on the Golden Gate Bridge that just happened recently. Yeah. So the thing is, it's effective at such tiny doses. You don't have to absorb that much into your body at all for it to start affecting you. You know, if you absorbed a little bit of morphine into your body through your skin, it would be irrelevant. It would just be, it wouldn't be enough to really be homeopathic. But with fentanyl, even absorbing a little bit into your body through whatever means can actually have a real effect on you that you're going to feel. Fentanyl is also used to cut opiates. Like a lot of times, like when people analyze, when you buy a little dime bag or something and they're like, what's actually in there? A lot of times people will add in fentanyl to make it stronger and make it, have more of a kick, more bang for your buck. And the danger is if somebody is used to shooting up like heroin or something, or Dilaudid or whatever they think they're shooting up, and somebody has actually cut it with fentanyl, it might be the actual dose that they need for the effect they want is way less than they are actually taking. Mm-hmm. So that's one of the problems. Again, if you're getting a drug and it's pharmaceutical, it's morphine or fentanyl or Dilaudid or whatever, and it's, USP, and, then you know exactly how much to inject for the effect that you want. But if you're just getting something from some dude on the street and he's telling you or she's telling you what it is, you don't really know what it is. And what's happened a lot is it's not exactly what people think it is. And often it's something more powerful. So the same amount that they think is going to give them a nice little respite from the world is actually enough to stop their breathing, and then they can die. So fentanyl is a big culprit in a lot of opiate overdoses because it's really common to cut kind of street opiates, even things that get sold as, oxycodone, like, oxys or something, sometimes are actually cut with, again, because it's cheap and it's powerful. So you can make a good profit off it. And just give anybody whatever they think they're getting. They want this, you just give them the fentanyl. They probably aren't going to know the difference. It's just a opiate escape. So fentanyl, but yeah, again, the thing that happened on the Golden Gate Bridge is probably just, if it's kind of hard to believe it was really just everywhere that they got it that easy, but I'd like to know more about how that happened. But it is administered as transdermal patches for sure. It's even administered as lollipops for kids. They have fentanyl pops. If you have a little kid coming in and you need to kind of get them anesthetized or at least kind of out of it for something, you can give, there are these prescription lollipops with fentanyl that you suck and will send you off. So.
For the case on the Golden Gate Bridge a couple days ago, or yesterday, day before last, it was my husband's coworker actually.
Oh, wow.
Yeah. He said he got there first, was feeling symptoms and had to call additional ambulance. He was actually responding to the guy crashing. And then a second police car came to help him out and he got exposed. And then a third policeman came out. So by this time, 3 or 4 came out, but they were all exposed. I think it was 6 of them.
You know what they were touching or how they actually, because it's not just going to be, unless there was a big bag and the whole air is just floating with it or something. I'm curious.
Because it was a crash site, they were all looking inside the car to see why he had crashed. And he was passed out. So I don't know if going in the car exposed them, but.
So they might, maybe as they were kind of sifting through the glove compartment or something, that might make sense. Yeah, no, that's scary. I mean, because you don't know what the dose is, and that one in particular, you can get into trouble really quick. So yeah, no, that's scary. All right, it is 9:42. So let's take a break. Let's return at 10:15. If you have, I'm looking here, for the RBC pre-labs, quite a few people have them done. Almost everybody has at least got the first try. There's actually, I have 2 people still with big pink things here saying like, you haven't even submitted as I started class this morning, which is really not a good sign. But if you have not submitted it or yours has comments and it says it needs to be revised and resubmitted, please do that before we start the lab. I would like everybody to be on the same page before we start the lab so you understand really clearly, what are we doing here? Why are we doing it? How do we interpret these results? Like, understanding what it means to see these different test tubes requires that you understand what the whole point of the whole experiment is. So please, we have like a half hour here. If you have not finished your pre-lab, please get that and get those ducks in a row. Otherwise, I'll see you all at 10:15.